Hello, everyone, and welcome to today's DDN seminar. I'm Luisa Torres, Assistant Science Editor for DDN, and I will be moderating our discussion. We have an exciting seminar planned for you today. Our speakers, Dawn Bowles, Samira Kiani, David Schaffer, and Wei-Dong Xiao, will discuss the major hurdles of AAV-based gene therapy, including inefficient vector delivery, immunogenicity, and viral vector integration. After the talks, our four speakers will participate in a live Q&A session. To submit your questions or comments, simply use the Q&A portal to the right of your screen, and we will try to get to as many questions as possible at the end of the seminar. I'd like to thank this opportunity to thank our seminar sponsors, VPS Bioscience, DNA Script, and Biored. Our sponsors have provided us with some helpful handouts related to today's webinar. You can find these in our handout section located on the right side of your screen, where you can also find your certificate of attendance for attending today's live event. As a thank you for registering for today's seminar, if you haven't already, please also check the handout section for printable phone and desktop backgrounds that have a fun gene therapy related design. And with that, let me introduce our first speaker, Dawn Bowles. Dawn Bowles is an assistant professor in surgery within the Division of Surgical Sciences at Duke University School of Medicine. She obtained her PhD in microbiology from Louisiana State University and received her postdoctoral training at the University of North Carolina, Chapel Hill. Her research team develops gene therapies to improve cardiac transplant outcomes, such as preventing post-transplantation complications and minimizing the need for immunosuppression. So let's take a minute to see Dr. Bowles' slides. And yeah, they're up. So Dr. Bowles, please take it away.
Thank you, Louisa. Thank you very much for the opportunity to provide an update on ongoing research in my lab. The talk is entitled Gene Therapy for Cardiac Transplantation Enabled by Ex Vivo Machine Perfusion. So today, I'm going to be talking about 3 of the 4 pillars that are required for successful gene therapy. In this instance, successful gene therapy for cardiac transplantation. I'm going to focus a lot on the delivery approach, which is normothermic ex vivo perfusion. I'm also going to talk about what we've done to develop a disease model to evaluate acute rejection for cardiac transplantation. And then I'll touch on some ongoing work that we're doing with therapeutic targets to minimize the need for chronic immunosuppressive drugs. First, for this audience, I thought I would give a little bit of information on the evolution of cardiac preservation. Historically, cardiac preservation has been cold static storage, which is basically what you see here, a heart in a plastic bag with some liquid solution in an ice chest. And this is not a very good thing. It limits the time that the heart can be out of the body to about less than 4 hours. And that limits the time or the travel time to get a donor organ to a recipient. And it also limits organ allocation. Recently, normothermic ex vivo perfusion has been utilized and has really evolutionized or revolutionized the realm of cardiac preservation. Shown here is the Transmedics device, or the organ care system, which is an FDA-approved device for heart preservation. And this enables the heart to be outside of the body for 8 or more hours. And this has really extended the distance that the hearts can travel, and it has expanded the organ allocation considerably. Creatively, we're doing a lot of different things are being done in the creative utilization of this device. In 2019, doctors at Duke made headlines by performing the first donation after circulatory death heart transplant in the United States, and they utilized this device, which enabled the transplantation of previously unusable hearts. And this has essentially expanded the donor pool by as much as 30%. So how does this work to enable gene therapy? And this is shown here, this video is shown here, a heart that is on the device. And this device maintains the donor heart in a near physiological beating state. And there is a perfusate that is recirculated on this device to the heart. I mean, this is based on donor blood, and it's supplemented with nutrients and oxygen in a very controlled and protected environment. And it's these traits, in addition to allowing multiple passes of the vector within the circuit, that maximizes opportunities for vector attachment and entry. So today I'm going to go over our experiences with two different vector types. First, adenoviral vectors, and then later adeno-associated viral vectors. Initial work, we used adenovirus because we knew that it worked very well on the heart. It transduces the heart very well as it provides a very quick onset of gene expression. And then later work, we're working with adeno-associated viral vectors, which are more clinically applicable, especially with the long-term gene expression. So this is our first foray into using this device with ex vivo gene therapy. And this was done, this was headed by Carmelo Milano, who is a cardiothoracic surgeon at Duke, and he is he's a partner. Our labs are partnered. This was also done by a surgical resident in the lab at the time, Moath Bashawi, and a cardiothoracic attending surgeon from Taiwan, Jineng Ron. This paper that was published in 2019 utilized the heterotopic heart transplant model. And the reason why we used a heterotopic heart transplant model, because it is an easier model to utilize in the sense that you're not relying on the native heart, or you're not relying on a heart to support life. So this is essentially a transplantation where a donor heart is implanted into the abdomen of a recipient. We also, in this study, utilized a cell saver to wash the donor blood because, not surprisingly, we found that there were components of the donor blood that were inhibitory to viral transduction. Then we applied to the circuit an adenoviral luciferase, an adenovirus that had CMV luciferase. And this was recirculated for a 2-hour time period. After that, the heart was implanted into the recipient animal, and we survived the animal for 5 days with no immunosuppression. What we found was very robust and diffuse luciferase activity and expression in the donor heart, with no evidence of luciferase protein or gene anywhere else in the recipient animal. This year, we've published our findings using the same system, but with an AAV vector. And this was done by 2 surgical residents in the lab, Yuting Chang and Michelle Mendio-Lacroix. Essentially, we have a viral vector. In this instance, it was also luciferase. We infused the vector into a port near the aortic root of the heart, and then perfused similarly for a 2-hour time period. And this enabled the vector to traffic to cardiac cells, and then potentially enabling the first steps in the transduction process. After the 2-hour time period, the transduced heart was implanted into a recipient animal in a heterotopic fashion, and then the animals were survived for 30 days on long-term immunosuppression. The results that we saw with AAV delivery on the organ care system was very robust and durable expression at 30 days post-delivery when we terminated the experiment. You can see here, these are results of luciferase activity in the allograft and the native heart, and we did 3 pegs of different doses, and you can see a dose-dependent effect on the luciferase activity. In this panel here with the stain, we stained for luciferase protein, And you can see very high levels of protein in the donor heart that received the vector in A and C, and no evidence of luciferase in the native heart in B and D. We also saw no evidence of allograft injury or off-target expression. In the top histology is the native heart, and the bottom histology is the donor heart. In addition, we saw no off-target effects. This is looking for luciferase expression in the livers of the 3 animals. And this is looking at the activity for luciferase activity in the lung, liver, spleen, and an area very close to where we implanted the heart. We also found that the perfusate composition could be tailored to enhance AAV transduction. We found components such as methylprednisolone and calcium gluconate could enhance AAV transduction. And we've also discovered additional perfusate additives that enhance AAV transduction and lead to significantly higher transgene expression. And we filed a patent application on that earlier this year. So now that we can show that this is a very efficient method for delivering viral vectors to a donor heart, what applications could we use this for? Shown here are 4 applications that we believe we would like to work with. The first one is improving the cardiac allograft outcomes. And you can imagine that you can provide protective genes that could confer traits to the donor heart that could counter rejection, could minimize ischemic reperfusion injury, or other adverse events of cardiac transplantation. And we've shown that it's very easily to deliver these vectors to the donor heart while in ex vivo perfusion. I'm sure everyone is aware of the recent last year transplantation done where a genetically modified pig heart was transplanted into a human recipient. And we believe that this could be utilized, ex vivo perfusion approaches could be utilized to improve xenograft outcomes, where vector genes could be delivered prior to transplantation of the pig heart into a human recipient that could boost transgene expression to protect against acute and subacute complications associated with xenotransplantation. But also the vector delivery can be used to deliver genes that are unable to be edited in the donor animal. A third application involves what one would consider the moonshot in transplantation, and this would be autotransplantation. And this is a concept where you could imagine that advanced heart failure could be treated by removal of the heart from the patient with advanced heart failure, placed onto this device, and then infused with different types of vectors that could reverse the damage and promote healing of the injured myocardium throughout the heart. Initially, this could be utilized in the same manner to correct genetic predispositions for advanced heart failure. Obviously, this is something to be considered very much in the long term. Another long-term application for this delivery involves the concept of organ banking, where we are moving towards having organs on a shelf that would be available, that would be stored and would be available when needed. You can imagine that you can perform short-term perfusion with viral vectors prior to some type of preservation, such as cryopreservation, using the same genes used in allotransplantation gene therapy. Or people are working towards longer-term perfusion, so perfusing longer than 8 hours, perfusing 24, 48 hours, long enough to enable gene expression to happen and to improve the function of the hearts prior to implantation. So the first thing that we're working on is to improve initial cardiac transplant outcomes. And we're focusing on trying to minimize the need for acute immunosuppression or long-term immunosuppression. In order to do that, we've been working for the past 3 years in the development of a cardiac rejection model in the pig. And this work has been done by Michelle Mendiela-Pla in the lab for the past 3 years. She's a surgical resident in the lab. What this model consists of is a Yucatan pig model of fully SLA mismatched animals, heterotopic heart transplantation, and then an immunosuppressive regimen of short term of 14 days, after which we remove the animal from we remove the immunosuppression from the animal, and then we observe for the development of rejection. In many different ways. We acquire serial blood. We have imaging modalities of MRI and echocardiography, as well as histological evaluation. And we essentially are looking for the heart to change into this very, very distressed heart. And I'm just going to go through some of the components of the rejection model that we've been working on. What we found that is very critical to the rejection model and having a very controlled acute rejection are two components. One is the degree of the SLA genotype mismatching, and the other is the immunosuppressive regimen. So what is SLA? SLA is like HLA. It's similar to the MHC system in humans. And there's a genomic organization of the SLA genes that are similar but a little bit different from HLAs. The blood from all of the potential animals that we are considering for transplantation studies are acquired and are genotyped by Lyndon Galt and Sam Ho at the Gift of Hope, where they develop these types of charts, and enables us to pick the suitable donor and recipient for our experimentation. This is the immunosuppression schedule that we have developed. It is a schedule that is a triple therapy of immunosuppression. It consists of a prednisolone taper, and MMF and tacrolimus for 14 days post-transplantation. In addition, the tacrolimus injections are started 3 days prior to transplantation, and we found that it is very critical to get a target trough level range of 5 to 15 nanograms per mil. And then after the immunosuppression is stopped at day 14, the allograft is monitored daily for the cessation of contractility. Other modalities that we use to evaluate the development of rejection consist of cardiac MRI, and this is done by Dave Wendell at Duke. And these are just some images that we've, these are some representative images. And we're using the cardiac MRI to gauge the development of the increase in LV myocardial mass in the allograft, as well as to characterize the interstitial edema and the development of fibrosis. And these are two parameters that are markedly increased in the rejected allografts. Another modality that we are developing is looking at the donor-derived cell-free DNA as a non-invasive way to monitor rejection. And essentially, you're just collecting cell-free DNA from the plasma and then performing sequencing to identify which reads correspond to donor and recipient SNPs. And then we can use this to monitor the donor DNA levels over time to detect the onset of rejection. We're still in the middle of doing this. We've performed whole genome sequencing on the majority of our pigs. And we are now in in the process of trying to develop a PCR-based panel of specific primers, which we could use to reliably determine donor-derived porcine sequences, as opposed to performing whole or shotgun sequencing of the donor-derived DNA. In addition, we have I say we, an interventional cardiologist at Duke, Murat Fudum, as well as a cardiac pathologist at Duke, has developed or helped us to develop the methodology to perform endomyocardial biopsy and histological assessment of the donor hearts. And this was recently published last year. But we're able to do this routinely, serially in these animals to obtain this biopsy information. This is showing that the rejection is a CD3, CD8 T cell infiltration at the time of the endpoint histology of rejection. And this is showing what it looks like from the T cell perspective of the rejected donor heart, where it's highly infiltrated with T cells, and then the native heart of the recipient, there's no T cell infiltration. In addition to that, we are performing immunophenotyping and have been working with Laura Hale, Kayla Parr, and Javed Mohammed, as well as Ji Lee, to identify the reagents and to develop the methodology to do the immunophenotyping of these pigs. And this is showing that as the rejection ensues, there's an increase in the number of CD8 positive T cells. So this has been an iterative process to develop this rejection model. Overall, we have determined that there is it's very controlled once we got the parameters down. So first, the first iteration at the top, we're showing the animals Rita and Roxanne, and these were partial mismatches with an immunosuppressive regimen of 21 days. And what we saw that there was no rejection. The endpoint of no contraction was never reached in these animals. And so our second iteration was much more aggressive. We went to a complete SLA mismatch, and we also used an immunosuppressive regimen that was much more aggressive of only 10 days. And what we saw in these two sets of animals was that we reached the endpoint very quickly within 17 and 22 days. And we saw a very severe rejection phenotype. We've since gotten to what we think is the sweet spot, where we are utilizing completely mismatched animals with an immunosuppressive regimen of 14 days. And in 6 of the 7 transplants shown here, we have an average time to rejection of 2 months plus or minus 1 month. And the final animal shown, Peggy, she never reached her endpoint despite these two critical components. But what we found that she was metabolizing the tacrolimus very differently and was always super therapeutic. So again, one of the critical parameters is to get the immunosuppressive within the 5 to 15 range in the trough. So now that we have demonstrated that we're able to efficiently deliver vectors to the donor heart using the ex vivo perfusion system, as well as a large animal model of acute rejection, what are we doing next? Well, we're focusing on trying to develop a gene therapy to induce tolerance in the cardiac transplantation patient. And we think that this is definitely an unmet need because of the influence of chronic immunosuppression, the negative influence of chronic immunosuppression on the recipient. There's a lot of malignancies, infections, and organ failures that are associated with long-term rejection. So our long-term use of anti-rejective medicines. Long-term, or our therapeutic hypothesis, is that we would be able to induce immune tolerance via AAV derived gene therapy delivered in an ex vivo fashion to express immune tolerizing molecules, and that these immune tolerizing molecules will reduce transplant failure, reduce or eliminate the need for immunosuppression, and potentially expand the donor pools for cardiac transplantation. So this is our pipeline for how we're evaluating our therapeutics. We're working with a cancer immunologist at Duke, Zach Hartman, and he has a nice model where we are evaluating our candidate molecules. He has a cancer model, basically, where in cancer cells, we're delivering the tolerance molecules and then implanting those cells into mice, and then evaluating the mice for development of tumor or no tumor. Those animals that do develop a tumor, those molecules then go into phase 2 of our pipeline, where we are utilizing a transgenic mouse transplant model, heterotopic heart transplant model, to evaluate the potential therapeutic targets before we move into the large animal preclinical model. The transgenic work, the heterotopic heart transplant experiments are being done by Jingwei Chen in the lab and Krish Dewan in the lab. Krish is a surgical resident, and Jingwei Chen is an attending cardiothoracic surgeon from Taiwan who's in the lab for the next year and a half. So this is just a diagram of the heterotopic heart transplant procedure in the mouse. It's essentially very similar to what we do in the pig, just at a very smaller scale. Krish and Jingwei have been spending the past few months learning and perfecting this technique. As you can see here, these are some examples of the successful experiments that they've done. This is how we are monitoring our mice. We have a donor, either a donor transgenic animal, or we're also using some viral vectors that express different targets. They're being implanted in these hearts from the donors, or being implanted into the recipients, and then we're monitoring daily by physical examination, by echocardiography, by PBMC profiling, similar to what we've shown with the pig. And we're also doing histological evaluation at the termination of the experiments. And these are some of the control experiments that Jingwei and Krish have been performing. These are just looking at control animals. This is not transgenic or not viral vector delivered animals. And this is just looking at the survival curves, whether you go from a BALB/C strain donor into a C57 Black 6 recipient, or vice versa. And basically, the model is a very consistent model. It takes about 7 days for all of the hearts to stop beating once post transplantation. Some key takeaway information from the histological assessments is that the rejection is characterized by marked cellular infiltration and loss of native myocardial architecture. And the histology can differentiate the loss of the graft from early ischemic causes versus immunological rejection, which is what we're looking at. Krish and Jingwei have been doing some pilot work with vectors using an AAV luciferase to work out any of the logistics around this before we delve into our therapeutic arm of the study. These are animals that were injected with an AAV luciferase by tail-vein injection, and you can see that the hearts do express this luciferase transgene. These animals are used as donors, and those donor hearts are implanted into the recipients in the abdomen, and you can see the imaging, the luciferase imaging here. The animal on the far left is a control animal that did not receive a vector, but whose heart was implanted in the same way. And you can see that you can visually see that these animals do express the luciferase in the donor heart. So finally, we are in the middle of looking and evaluating our first 2 therapeutic candidates in this mouse model. The data I don't have today to show you, to be determined. But then just to remind you where we are, we're in phase 2 here, and we're hoping to move at least 1 therapeutic candidate into phase 3 to evaluate in our rejection model sometime later this year or early next year. And the ultimate goal is that we would want the hearts to look like the hearts on the left as opposed to the heart on the right. The heart on the right is a fully rejected heart, and the heart on the left is a not rejected heart. So I just want to provide you with a little bit of the literature that we've been developing on gene therapy for cardiac transplantation. We've recently been presenting at numerous scientific sessions. And there's also a Frontiers in Cardiovascular Medicine Graft Preservation topic that I am a special editor for, and we are still receiving or accepting articles for this collection. And finally, in closing, I would like to thank the individuals and the entities who have funded this work. And thank you very much.
Thank you, Dr. Balls. Now we'll go with our second speaker, Dr. Samira Kiani. Samira Kiani is an associate professor of pathology and bioengineering at the University of Pittsburgh, working on strategies for safe gene therapies. She earned a medical doctorate from the Tehran University of Medical Sciences and completed her postdoctoral training at the Center for Synthetic Biology at the Massachusetts Institute of Technology, where she worked on reprogramming the function and behavior of mammalian cells using CRISPR-Cas9 technology. She's the co-founder and CEO of GenXGen, a synthetic biology startup focused on building gene therapies to control immunity. And
thank you very much. I'm just going to share my screen.
Yeah. Go ahead and share your slides.
Do you see it?
Not yet. It's loading. Yeah, we can see it now. Oh, yeah, go ahead. Thank you.
Well, thank you for the opportunity to be here. I'm here basically wearing 2 hats. Part of my position right now is at the University of Pittsburgh, where I run a research lab and applying synthetic biology to address problems associated with overactive immune response using also CRISPR epigenetic editing platform. But also as part of my other, capacity, I have started a company called GenXGen. So the presentation that I will be having today will be mostly, coming from my role at GenXGen, and hopefully we'll be able to describe to you some of the work that we are planning on doing at GenXGen to address the immune response of gene therapies. So I want to start by saying that, as all of us know, that gene therapies bear tremendous potential in addressing a lot of different genetic and acquired diseases. And we see a number of these gene therapies that are active in clinical trial, and some of them also have received FDA approval. There's a lot of opportunities for growth in the field of gene therapy. It is expected to reach about or more than $60 billion market opportunity by 2030. And in most of these gene therapy trials that has been done in clinical trials or even the FDA approved one, majority of these utilize viral vessels such as adeno-associated virus to deliver gene to the patients. And in parallel, increasing research and innovation in the field of gene editing or epigenetic editing tools, such as CRISPR or zinc finger technology, has had huge impact on opportunities and market size for the gene therapy, with about 2,800 gene and cell therapy products in the market during development. So there is a lot of potential there. However, safety and efficacy challenges hamper the success of gene therapy. What do I mean by that? There are, the idea is we put foreign genetic element using, for instance, viral vessels into the patient's body. So body's immune system recognize these as foreign and form immune response. And there are two types of immune response. Some patients already have exposures to these viral gene therapy, viral vessels for gene therapies, including adeno-associated virus, and they have already formed immune response. There are pre-existing antibodies, and this is called pre-existing immunity. And there's immune response that is formed when body sees the viruses for the first time, and that's the new immune response that is formed. So both of these instances provide challenges for the success of gene therapies. As such, about 40 to 50% of total patients that can otherwise be included in gene therapy trials are excluded because of circulating antibodies that recognize these viruses and disable them. And because of the new immune response that many people form after they receive the first dose of gene therapy, the redosing will become an issue. There are many examples that these immune response or toxicity that associated with gene therapy viruses happen in clinic, and there are gold standards to suppress the immune response against gene therapy, including corticosteroids. But unfortunately, corticosteroids are generally ineffective in controlling this immune response, and there's also an issue to sustain the expression of the transgene from the viral vector that is delivered to patients. For instance, in one example, in hemophilia trial, the patients receive corticosteroids, but even under the corticosteroids, these patients developed toxicity and they lost the transgene expression. So there's always this battle: what is the correct and efficient immunosuppressive regimen that we can use? And most of the current one generally create inconsistent result in clinic. And we see many examples of these, battle with the immune response to gene therapy that is currently happening in clinical trials and, in clinic. Most of the time, patients with high neutralizing antibodies are actually excluded from trial. In about 45% of trial, as you can see, there's neutralizing antibody exclusion criteria that you can see if there's high antibody in the patients that are excluded. And there are examples of failure of gene therapy trial and massive loss of capital that exists in industry because of the toxicity and the problem of immune response to gene therapies. So I hope that by these introduction, I have convinced you that immune response to gene therapy creates massive burden both on the healthcare system and also the patient themselves. In the patients, they create adverse effect They decrease the treatment options. In many cases, also they delay treatment. But on the healthcare system, there's a massive amount of cost and lost resources that goes into developing these gene therapies that ultimately can fail because of the toxicity. So there is a lot of work that has been done in order to address these. And I have listed some of these work, and this list obviously is not complete because this is a very active area of research and exploration to be able to address this problem of new or pre-existing immunity to gene therapies. And they range from simple modifications in the protocol, including just administering higher dose of virus to the patient to overcome the pre-existing immunity or that exists in the patients, or use empty capsid as decoy to absorb and remove the existing antibody. But all of these still lack the capacity to provide consistent result in clinic. There are other approaches such as modifying AAV capsid or using different AAV serotype each time that you need to redose AAV. But this is also technically challenging and can be expensive, and you cannot really scale it up. And while at the same time think about the cost of therapy. There's a lot of work that has been done in improving manufacturing pipeline to reduce the immunogenicity of viruses, to make sure that they are pure, they don't carry any potential contaminants that can activate the immune response. And obviously, the gold standard has also been creating and providing a regimen of immunosuppressive drugs that can control the immune response. But these, as I mentioned, their effect cannot not last long. They have massive side effect, and the result in clinic hasn't been consistent. So what we have been trying to do is to tackle this problem, both the problem of new or pre-existing immunity, but also develop a strategy that can be applied not only in the field of gene therapy, but across different fields in which controlling the immune system is important, including transplantation, allergy, and beyond, while at the same time being considerate about the cost as well as the scalability of the approach that we are creating. So in order to tackle this problem, we focused on some lessons from vaccinology, the field of vaccine itself. Within the normal vaccine, and we're all familiar with COVID vaccine, you introduce an antigen to the body, whether this is in the form of protein or other forms, like previously even killed viruses. You introduce the antigen to the body. This antigen is picked up by cells, professional cells that are called antigen-presenting cells. Dendritic cells, macrophages, are one of those, are among those antigen-presenting cells. Once the antigen is picked up by the antigen-presenting cells, it's processed and presented at the surface. It requires additional signals to activate the immune response. In vaccines, there are adjuvants that are incorporated in the formulation of that gene, that they play the role of, providing that additional stimulation for the immune system. And that's how the immune system evolves to recognize and remove the antigen. But we can go a reverse path. If at the same time that we produce or we introduce antigen to the body, we introduce signals that inhibit the immune system or remove the signals that activate the immune system, we can train the immune system to ignore the antigen or create tolerance against that antigen. So we call this reverse vaccination or tolerogenic vaccination. And for simplicity, those signals that inhibit the immune response within, produced by the antigen-presenting cells or provided in the formulation of the reverse vaccine can be called anti-adjuvant. And the concept of reverse vaccination has been used in various disease conditions to induce tolerance against specific antigens. We have seen that in the context of autoimmune diseases or transplant rejections, or even in the field of gene therapy to induce tolerance against, the transgene that has been delivered, like factor 8 or 9 in hemophilia. But most of the time, these tolerogenic vaccines that have historically been produced rely on production of proteins. So you create peptides and deliver peptides to the patients. Most of the time, it's hard to define what is the antigenic source, specifically in complex diseases such as autoimmune diseases and transplant rejection. And therefore, the field of tolerogenic vaccine has, despite its great potential, has remained to show its fullest potential in clinical trial, partly because larger scale manufacturing of these peptides are sometimes very challenging. You don't know what's the right antigen to introduce to the patients and so on. So the field is poised for innovation. We wanted to really also, tackle these challenges that exist in the field of reverse vaccination, because we were interested to see how we can take this concept into advancing the tolerance against gene therapies. So in order to do that, we went back into how the mechanisms of adaptive immune system is and how it is informed by the innate immune system sensing of pathogens through our innate immune system. So let me explain to you what I exactly mean. So as I said, when these professional antigen presenting cells, such as dendritic cells, see antigens, and they pick up the antigens, they process the antigen and they present them in the MHC on their surface of these, cells. But at the same time, in order for them to undergo the full process of maturation, they need additional immunostimulatory cues. So they need co-stimulation in order to be able to become fully mature, to be able to activate the immune system, to create cytotoxic T cells or T helper cells. And there are two types of cues that can activate them. There are some, co-stimulatory cues, as well as some cytokines that is produced by these cells, including, I've listed these, IL-1, IL-6, IL-8, and these cytokines that are, they're connected and associated with activation of the immune system. If these immature DCs, when they see the cell, when they uptake the antigen and they see the T cells, at the same time, they do not create these immunostimulatory cue, but they create immunosuppressive cue, the tolerance happens. So now, these immunostimulatory cues are mostly developed through the function, or developed and produced through the function of innate immune system. So let me explain to you this other graph. When the cells see a pathogen, including a virus, there are certain pattern recognition molecules, including toll-like receptors, inside these cells that can recognize foreign component of a virus. For instance, they can see the DNA of the virus. They can sense the capsid of the virus. And following that, they can activate some downstream molecules that can inform the cells to produce the cytokines. And one of these very important molecules that sit in the intersection of sensing and then production of these cytokines is MyD88. MyD88 is very important. This is the molecule in yellow that you can see, in transmitting the signals from these pattern recognition molecules into the cells. And ultimately, the cells activate, the MyD88 leads the cells to activate the NF-κB pathway and form a lot of these immunostimulatory cues, a lot of these cytokines. So we were very interested to see if we target MyD88, can we actually create a new anti-adjuvant strategy to silence the antigen-presenting cells and deviate their differentiation away from, activating or mature DC into tolerogenic disease. So the question was, can we develop novel anti-adjuvant and tolerogenic vaccines against gene therapy by looking at the MyD88? And we were particularly interested in MyD88 because it is kind of a Swiss Army knife. There's a lot of potential or connections of MyD88 with disease conditions, including cancers that somehow are formed with overactivity of MyD88. There are a lot of cancers, and MyD88 plays a key role in oncology. But also importantly, in this case, is the important role of MyD88 in the activation of the immune system, in driving the adaptive immunity. And it's interesting to see that many vaccine adjuvants also function through MyD88 activation. So we wanted to be able to silence these MyD88 gene, but current approaches to target MyD88 have failed to enter a clinical trial, mostly because these either small molecule inhibitors or siRNA-based inhibitors are not specific. And because of the lack of specificity, they had a lot of toxicity. So we turned into other approaches, including epigenetic editing, in order to control MyD88. As you know, epigenome regulates volume of gene products by determining how tight or loose DNA is packaged within the cells. And there are advances in the field of epigenetic editing recently because of the new DNA targeting modalities, such as CRISPR or zinc fingers, that have been created and produced. There's a possibility to create targeted epigenome editing by fusing these DNA targeting modalities, such as CRISPR or zinc fingers, to chromatin regulator, to gene regulators, and be able to specifically target a gene and regulate the expression. So we have a lot of work done in this area to develop targeted epigenetic editors for a variety of genes. And we think that there are unique advantages, including the fact that it can be more safer, and you can tune the level of the critical genes without disrupting them. So understanding that epigenetic editing can be a very interesting way and effective way to regulate genes such as MyD88, we developed a platform technology that combines mRNA-based strategy, targeted gene regulatory strategy to specifically address the immune response against gene therapy. Our platform, as I said, is an mRNA-based targeted gene regulatory therapy that specifically regulates the volume of the genes that derive an unwanted immune response. And the reason we are focusing on mRNA is mostly because of the cost and the scalability of that. The core of the platform that we are developing is a programmable immune regulator rationally designed by combining nanoparticle technology, mRNA, and epigenetic editing to regulate the immune system. We call it UniGex. In other words, UniGex is a tolerogenic chassis rationally designed to be providing us the anti-immunity capacity. It composed of a nanoparticle and mRNAs that encode different, tolerogenic products and other enhancers that would allow us to enhance our tolerogenic capacity. So it's a multi-component chassis that keeps the immune cells from activation. And as I mentioned, our first focus is to regulate MyD88 because it is a key gene that drives the effective immune response against the antigens, and specifically viral-based gene therapy antigens. We have done a number of studies before that using the CRISPR technologies that we have shown that we can target the promoter region of MyD88 and create an epigenetic repression and tune down the level of the MyD88. Our prior approach, which is published, relies on basically combining Cas9 protein with potent epigenetic regulators that we have already published. Those that are combination of different chromatin domains that are fused together. In this case, HP1A and CRAB domain that we bring in to the mix and we use for epigenetic editing. We have used mouse to examine the effect of MyD88 repression using the CRISPR in forming the humoral response against AAV. In our case, we have in the published paper, we packaged CRISPR within AAV1 and we injected these into mice. The AAV1, as I said, produced CRISPR that represses MyD88. And we looked at the AAV1 specific antibodies in these mice about 3 weeks after injection. And we show that MyD88 repression, when it's incorporated within AAV1, decreases the antibody response against AAV. And that redosing also, when there is already a repressed MyD88 status, allow us to control the further rise in antibody. And so we have tested these in the context of other, AAV serotype and asked whether, suppression of MyD88 when it's incorporated within AAV1 can control immune response against AAV9, for instance, and allow redosing. And, the answer is that yes, if you suppress MyD88, even if it's within AAV1, you can actually control, for instance, in here, antibody response against AAV9 as well, and allow redosing, and you achieve higher transgene level upon redosing. So in this case, the mechanism has been because the AAV vector that gets into the antigen presenting cells at the same time produces MyD88 suppressor. We believe that these spatio-temporal confinement within the cells allow us to deviate the immune recognition to the tolerance, and that's the mechanism of action. But in order to translate these, AAV-based approach to clinic, we faced a number of challenges, including limited packaging capacity of AAV, high cost of GMP manufacturing of AAV, and difficulty to scale. But also, really, we don't want to, use this AAV delivery to control the immune response to AAV when there is, pre-existing immunity to AAV. So we want other ways to actually translate this idea of a novel anti-adjuvant based therapy and vaccination therapy to clinic. And so therefore, we turn our attention into mRNA-based vaccination. So now we have the mRNA that combines with nanoparticle and epigenetic editing of MyD88, which was our anti-adjuvant, combined with the antigen of interest, in this case, AAV, for instance, coat protein, that now can allow us to achieve antigen-specific immune tolerance. So the idea would be to use a lipid nanoparticle, incorporate our anti-adjuvant, which is an mRNA that encodes for MyD88 suppressor, and another antigen that encodes for another mRNA that encodes for antigen we want to tolerize against. In this case, for instance, viral capsid protein. And there are certain design considerations we have had, including the fact that in all of these, the therapeutic regimen or the vaccination needs to be immunosuppressed. Therefore, lipid nanoparticles need to get into immune cells and be immunologically silent as much as possible. mRNA needs to be designed in a way to be immunologically silent and be expressed effectively. And we might need to have right antigen and Uniject, which is our anti-adjuvant combination. So we've done a lot of, iteration of these. We've worked a lot to actually make this vaccine. We shifted from CRISPR to zinc finger because we think that zinc fingers can be better suited for this purpose. They're more from eukaryotic origin. They have less of immunogenicity as compared to CRISPR. And it's also a single component. You are not dealing with a Cas9 and guide RNA. So we have tested a number of zinc fingers that repress the MyD88, and we've selected one that works very well. And then we have packaged this in the form of mRNA into lipid nanoparticle and injected lipid nanoparticle in vivo to show that we can actually suppress MyD88 in vivo. So the experiment is LNP injection systemically. 6 hours later, we harvest the organ, and in this case, in the spleen, we have seen that LNPs themselves can actually activate MyD88 somehow. But when you add zinc finger repressor that suppresses MyD88, you decrease the level of the MyD88 into uninjected control. So that's a very good kind of like strategy to keep our chassis immunosilenced. And then we have also shown that in the context of heightened inflammatory state, such as induction of LPS-based septicemia in mouse, if we inject these mice with our Unigex chassis, which combines the LNP and mRNA that suppresses MyD88, we can actually control the MyD88 level, similar to animals that are in a non-septicemic step. So this is the septicemia animals. You see the MyD88 goes up, and then when you inject these LNPs that carry the MyD88 suppressor, you decrease the MyD88 in blood and lung. So this is further validation that our chassis is immunosilenced and that control MyD88. And we have also shown that this strategy can also control other cytokines that are important in activation of the immune response as compared to the control that didn't receive the MyD88. So therefore, we design our experiment, which is our standalone vaccination strategies, in which we induce these mice to develop the antibody response against AAV by 2 injections of AAV, in this case AAV to mCherry, to induce antibody response against AAV, and then we provide a standalone vaccination using these LNPs together with the antigen that we want to tolerize against. And then after 2 vaccination, we subject these mice again with another dose of AAV and wait a few weeks to sacrifice the mice and look at the level of AAV specific antibodies. These are very preliminary data that I'm showing you. Obviously, we need to repeat these and increase the robustness. But what we have found is that in the case of pre-existing immunity, tolerizing with these vaccination allow us to decrease AAV to specific antibodies and achieve higher level of transgene, in this case, the AAV that was injected after the tolerization in a spleen and lung. And also we see a trend of higher level of AAV DNA in these mice. Also, we would show that the level of some of these pro-inflammatory cytokines that activate the immune system are actually lower. There's a trend to decrease this level. Obviously, these are preliminary studies that we are doing, but hint to the possibility that these type of vaccination can be used specifically in the cases that we have pre-existing immunity. And there are a lot of competition, and I know that I'm going over time, but 2 more slides that I just want to highlight, that there are a lot of work in parallel to ours that are being done in order to innovate in this space. And one of the 2 works that I want to highlight is the work that has been done by Selecta, that they package rapamycin, which is mTOR inhibitor, within the nanoparticle, and they have used this with AAV-based gene therapy to control the immune response. This approach has been shown to be effective when applied with the virus, but also has a lot of side effects because of rapamycin being very potent immunosuppressor. What we believe this approach can help us achieve is that it is mRNA-based epigenetic editor allow us to tune the expression of the gene, in this case MYD88, but not hammer it down, and therefore provide a safer margin. mRNA is cheaper and scalable, and therefore we can potentially decrease the cost as compared to, for instance, AAV-based delivery of these, approaches. But also the ability to develop reverse vaccine by introducing the antigen we want to tolerize against provide us an opportunity to be able to specifically control and tolerize against antigen of interest that can expand from different gene therapy vehicles, including AAV, adenovirus, HSV, and so on, but also get into gene therapy cargos such as, hemophilia. And also it can control the immune response in other instances, including cell therapies, transplantation, and beyond. So I just want to conclude by just summarizing that we are developing tolerogenic vaccines for gene therapy viruses by combining mRNA technology, epigenetic editing, and lipid nanoparticles to create a tolerogenic or anti-adjuvant chassis that they can carry the antigen of interest to then train the immune system to ignore these antigens. And with this, I want to also kind of acknowledge my collaborators, Ibrahim Khani Lab and Catherine Whitehead Lab, who actually help us develop these nanoparticles as well as these in vivo testing, and the funding organization that have allowed this work to advance in my lab and get into GenXGen. Thank you very much.
Thank you so much, Dr. Kiani. I'll now introduce our third speaker, Dr. David Schaffer. David Schaffer is a Hubbard Howe Professor of Chemical and Biomolecular Engineering, Bioengineering, and Molecular and Cell Biology at the University of California, Berkeley. He received a PhD in Chemical Engineering from the Massachusetts Institute of Technology and conducted a postdoctoral fellowship at the Salk Institute for Biological Studies. In his current position, he applies engineering principles to optimize gene and stem cell therapies. He uses directed evolution to engineer targeted and efficient viral gene therapy vectors, as well as new technologies to investigate and control stem cell fate decisions. He co-founded several companies, including 4D Molecular Therapeutics, Ignite Immunotherapies, and Rewrite. So let's see your slides.
Alrighty. Is this coming across okay?
Yes.
Great.
I just need to, yeah, that's great. Yeah. Okay. So please take it away. Thank you.
Thank you very much for the invitation and the chance to share some of our work. So as was mentioned, we work on both cell therapies as well as gene therapies, but the latter is going to be my focus today. And specifically, we've been working for now almost 25 years, about 25 years on adeno-associated virus. And of course, the field is very familiar with AAV because it's been broadly used in a number of attempted clinical trial development pathways. So we know the attributes of AAV, specifically the cap encodes several structural proteins, 60 copies of which will self-assemble to form a hollow icosahedron, and then a single-stranded genome containing, in this case, a recombinant genome that has a gene of interest in it, will snake its way into the interior of the particle to be able to create an infectious variant. These variants have a number of properties that are advantageous for human trial use, such as the fact that we're starting from a relatively safe place in that 90% of the human population has been exposed to one or more serotypes naturally of AAV, and it's non-pathogenic. It's been utilized in a couple hundred clinical trials involving delivery to muscle, liver, lung, brain, retina, and other tissues. Upon reaching the nucleus of a target cell, AAV deposits its single-stranded genome, which then converts into transcriptionally active double-stranded DNA and can persist for years or potentially even decades. We'll see as time goes on with human trials. The particles are manufacturable. There needs to be improvements in this to be able to reduce costs and increase scale, but they're sufficiently manufacturable now to be able to result in 4 FDA-approved gene therapies, including the first in 2017, which was Luxturna by Roche for the treatment of Leber congenital amaurosis type 2, Zolgensma from Novartis for the treatment of spinal muscular atrophy type 1, Hemgenix, which happened last winter, from Unicure for the treatment of hemophilia B, and Elevidys just last week from Sarepta, which was approved for the treatment of Duchenne muscular dystrophy. So there's a lot of momentum within the field, but there are also challenges that are going to preclude us from extending these clinical successes to the majority of human disease. So, for example, the first of these is one that we just heard a lot of terrific ideas and data about, which is pre-existing antibodies or pre-existing immune responses, where patients, most of us, harbor high titers of antibodies directed against the natural versions of AAV. In addition, AAV tends to biodistribute to the wrong tissues in vivo. So, for example, with an intravenous injection, the vast majority of AAV will go to the liver. It's not particularly efficient at being able to transduce hepatocytes within the liver. And furthermore, it doesn't typically reach other tissues of therapeutic interest, such as skeletal muscle or heart. It inefficiently spreads within that target tissue. It has the natural versions currently have zero ability to target delivery to some cell types and not others. It's inefficiently taken up into most target cells. It's a respiratory virus by evolutionary origin, of course. And it's, as we discussed, challenging to manufacture at high scale. So you might ask the question, as a result of all these challenges, why do we already have 4 FDA-approved products? And within these clinical studies, investigators really undertook Herculean efforts to be able to overcome these shortcomings with AAV and get the efficacy that they did observe within those trials. So, for example, 3 of these products, Zolgensma, Hemgenix, and Elevidys, require very high doses to overcome the intrinsic poor or low delivery efficiency of AAV. And Luxturna, as we'll talk about in a minute, requires a very invasive surgery to be able to put the AAV right next to the surface of a target cell, because otherwise it would have had no hope of reaching that target. So it's possible in some cases to compensate for poor delivery to some tissues, such as heart or skeletal muscle, by simply increasing the dosage. For example, this approach has been used in a number of trials, including that of Astellas, which was trying to deliver AAV8 to the heart. And to compensate for the fact that the vast majority of AAV8, when injected intravenously, goes to liver rather than heart, they increased the dosage to patients up to the point where it was over 100 AAV particles per every single cell within the body. So AAV is a very stealthy virus. It's non-pathogenic. But if you get to the stage where you're delivering 65 milligrams of antigen to a patient, the immune system unfortunately begins to notice this. And there were very, unfortunately well-known fatalities. 4 boys were lost in that clinical trial as a result of immune responses in the liver. So the bottom line is that the field is really beginning to see some success and patients are benefiting, but viruses, of course, never evolved in nature for our convenience to use as medicine. So it's up to us to re-optimize these at the molecular level to make them better for our purposes, rather than having to contend with the disadvantages of the natural versions of AAV. So this is a very challenging undertaking because unlike a small molecule that's 1,000 molecular weight, or perhaps an antibody where you're engineering the complementarity determining region, which is maybe a few thousand in molecular weight, AAV is a 4 million molecular weight particle that contains both protein as well as nucleic acid within it. It's produced in a complex bioreactor system. It has to, upon injection, biodistribute to the right tissues, such as the heart, extravasate, be able to penetrate deep into those tissues, bind to a cell surface receptor, undergo receptor-mediated endocytosis, escape the endosome, traffic to the nucleus, and finally release its genome, which itself is also a complex drug product because it has regulatory as well as coding information inside of it. So viruses, in other words, are very complicated, and it's difficult to re-engineer these molecules at the molecular level to be able to target them towards any given cell or tissue within the body. So we began to, 25 years ago in my lab, apply another approach than what was being worked on at the time. Up until that stage, people were hypothesizing which regions of the virus should be changed and introducing point mutation by point mutation to see which modifications of the virus could improve efficiency. So we began to take a higher throughput approach that was based on some technologies that were being developed in the enzyme engineering field back early in the 1990s, and specifically the concept of directed evolution. So I'll go through this quickly because this is this is something that's being used within the field. So you start with a wild type capsid. None of these are optimal for therapeutic delivery inside the body of a DNA medicine, but each one is an interesting starting point for further engineering. So we genetically diversify these using a variety of different approaches, everything from error-prone PCR to saturation mutagenesis of key surface loops to the generation of chimeras. In the past 7 years, we've been using computational approaches to develop better libraries, including ancestral reconstructions, as well as machine learning. But the net result is we now have about 45 individual libraries in which diversity has been introduced into the capsid in a variety of different ways. Each one of these is around 10 to 100 million in size. So in sum total, we have over a billion new engineered versions of AAV, any of which may be a solution to a delivery problem that we're trying to solve. So for example, let's say this variant over here in the dotted red line is the optimal variant to be able to reach the heart after an intravenous administration. Each one of these particles is now composed of a variant capsid surrounding the gene, the cap gene that encodes that capsid. So this particle contains inside of it the sequence of the cap gene that results in optimal delivery. So we can perform iterative selection where we do an injection IV into an animal model, recover the heart tissue, amplify the cap gene that made it there, repackage, readminister, and go through these successive rounds of selection until this large library converges down to a small number of variants that now have the properties of interest. We initially did this work in cell culture in the 2000s, about 10 to 15 years ago, started to do work in mouse, but had progressively been moving towards larger animal models as we want this to be primed to enter into the clinic. So today I'm going to be talking about work in mouse, old proof of concept work, as well as work in non-human primate and then in human primate. So I'd like to start with the retina, since the retina, of course, was the tissue in which the very first AAV was FDA approved, and it's an important clinical target. So the structure of the retina is that the lens focuses light onto the back of the eye, and the back of the eye contains a number of layers of neurons. All the way at the back is photoreceptor neurons. When they get hit with a photon, they send an action potential to several layers of interneurons that process that visual information. And finally, ganglion cells have axons that bundle together to comprise the optic nerve, which then conveys that information into the brain for further processing and interpretation. Unfortunately, mutations in over 200 genes can kill off photoreceptor neurons, robbing patients of their vision. And in addition to these rare disorders, there are also very large patient disorders, unfortunately, such as age-related macular degeneration that kill off photoreceptor neurons and rob people of sight. So one would like to deliver a therapeutic to be able to treat this condition. And the best way to deliver a drug into the eye is through an intravitreal administration, an injection into the vitreous fluid of the eye. And this is actually a very safe and well-tolerated route of administration. It's conducted millions of times a year across the United States for patients suffering from wet age-related macular degeneration. In that condition, these patients have aberrantly high levels of vascular endothelial growth factor within their retina, and that VEGF stimulates the ingrowth of blood vessels into the retina in a way that, for reasons that are not completely understood yet, kill off photoreceptor neurons. So there are a number of protein therapies, including Eylea, which neutralize vascular endothelial growth factor, and clinical ophthalmologists will inject any given patient with on the order of 10 injections per year with this anti-VEGF therapeutic. And it's very well tolerated and safe, and a clinical ophthalmologist can do 50 of these in a morning. Unfortunately, AAVs don't work the same way as smaller anti-VEGF proteins, and several hundred microns of dense tissue lie between the vitreous fluid of the eye and the photoreceptor neurons, and this prevents natural versions of AAV from making the trip from the vitreous to the target cells, the photoreceptors. As a result, investigators in that FDA-approved product had to invent a new approach for surgery a subretinal surgery where the needle is pushed all the way through the vitreous fluid, used to puncture the retina and to inject the virus-containing fluid behind the photoreceptor neurons. This is problematic for a couple of reasons. It adds insult to injury to patients already undergoing a retinal degenerative disorder. It damages the retina and can result in loss of vision in a way that is not, the virus doesn't cause this, even injection of saline can do so. And furthermore, it only exposes around 5 to 10% of the surface area to the virus. And the remainder of the surface area, the retina, progresses in disease. So a number of years ago, we began to work on the problem of isolating AAVs that, when injected into the vitreous of the eye, could reach the photoreceptor neurons. This was initially done as a proof of concept within mouse, in which we used a mouse strain in which GFP was selectively expressed within photoreceptors, injected the library into the vitreous of the eye, waited a couple of weeks, used fluorescence-activated cell sorting to pull out the photoreceptor neurons, PCR amplified the AAV genomes that made it there, and went around that loop about a half dozen times until, at the time, this is a small library. We only had 10 million back in 2010 or so. But the small library converged down to just a handful of variants, including one dominant variant that contained a specific peptide inserted into the tip of the threefold axis of symmetry, along with some point mutations on the surface of the virus. We loaded this variant with GFP to test its efficiency. Shown here is the parental serotype AAV2, which is actually used in Luxturna, that FDA-approved therapy. And you can see why they have to do the subretinal surgery, because if you introduce AAV2 into the vitreous, in this case in a mouse, you can see it barely touches the retina, hits some Mueller cells, some ganglion cells, but doesn't reach the outer nuclear layer, doesn't reach the photoreceptors. In contrast, the variant that we created, which we called 7M8, is able to penetrate deep into the retina and to infect essentially every single cell type along the way. And furthermore, we're not limited to 5 to 10% of the surface area of the retina. This transduces the entire retina. Okay, so we elucidated the mechanism for this, and in particular, AAV2, which is the parent to this virus, binds to heparin sulfate as its primary receptor. And there's a lot of heparin sulfate at this interface between the vitreous and the neuroretina. There's this barrier called the inner limiting membrane that contains a number of extracellular matrix proteins and proteoglycans, including heparin sulfate. So AAV, because it has 20 binding sites for heparin sulfate as a result of its icosahedral geometry, binds to the inner limiting membrane like glue and has no opportunity to penetrate deeper into the retina. So the variant that we created actually has lower affinity or avidity for heparin sulfate, so it transports through the tissue better. And furthermore, it has the capacity to bind to a novel receptor whose identity we've not yet cloned. Hey, we're very interested in translating this work to the clinic. This study, as well as a number of others, convinced us that the technology was working and was ready for clinical translation. So to do so, I co-founded a company back in 2013, and this company is located about 3 to 4 miles from the UC Berkeley campus, called 4D Molecular Therapeutics. The company has now accessed the public markets so that it's been able to fundraise to be able to head into clinical development. So we have 150 employees, are working primarily in 3 therapeutic areas, the retina and the lung, as well as the heart, and have 5 ongoing human clinical trials now within those 3 therapeutic areas. And I'm going to talk about 2 of those in particular. So there's a saying in the directed evolution field, which is that evolution teaches you get what you select for. So that variant that we selected in mouse, we attempted to utilize it within a non-human primate. And not surprisingly, the structure, the volume, the size, as well as the molecular biology or cell biology of the primate retina is different from that of the mouse. And that 7M8 variant evolved in mouse doesn't work very well in primate. So we went back to the drawing board, and now with our improved libraries and over 1 billion variants as a starting point, we re-evolved a variant that was capable of reaching the outer retina from the vitreous within a non-human primate. And this resulting variant was actually different from the mouse variant considerably. We began to test it, again, starting out with GFP as a marker. And shown here are a couple of negative controls. First, if you inject AAV2 into the vitreous, it's able to transduce a small number of retinal ganglion cells. Specifically in the center of the retina, there's a region called the macula, where that barrier, that interlimiting membrane barrier of ECM and proteoglycan thins out. So AAVs begin to access a superficial little ring of retinal ganglion cells, but the AAV doesn't penetrate any further and certainly doesn't reach photoreceptor neurons. You can get AAV2 to reach photoreceptor neurons by doing a subretinal surgery, but again, you can't control the shape or the size of this region, and it's only a small percentage of the surface area of the retina. When we inject this novel variant R100 into the vitreous of a non-human primate, we began to see very high levels of expression. Within 1 week, you begin to see those retinal ganglion cells, but then transduction begins to spread across the entire retina. And we performed literally thousands of histological sections within these primates and did immunostaining as well as native GFP fluorescence imaging, and saw GFP expression across the entire retina, regardless of whether it was in a very bright region or a lighter gray region. And this appears lighter gray simply because the camera on the microscope is auto-adjusting its exposure to enable dynamic range in GFP expression to be visualized. So I'll note as well, this is a 6-month in-life study. The animals were not immunosuppressed the majority of the time. So they were expressing high levels of a jellyfish protein in the absence of immunosuppression, indicating that the vector appeared to be safe so far. So based on this momentum, we began to carry this vector into human clinical trials. The first 2 trials were dosed in May of 2020, were delayed a little bit as a result of COVID. I'm not going to be talking about those results. All of these results I'm discussing today have been publicly presented, and those can be found on 4D's website. But initially, we were treating orphan indications, choroideremia and X-linked retinitis pigmentosa, which kill off rod photoreceptors initially and then culminate with the loss of cone photoreceptors as patients go blind. So this vector carrying the transgene that's mutated within either of those conditions appeared to result in a slowing down in the degeneration of the retina within the late stage patients that could be enrolled in the trial by the FDA. And as a result of that, those trials are progressing towards later stage trials. But this study I want to talk to you about today is one that I'm especially excited about, because it's not an orphan indication. It affects 1 million Americans, and this is age-related macular degeneration. So specifically, as I mentioned earlier, there are protein therapies that are routinely used to be able to treat wet AMD, such as aflibercept, whose commercial name is Eylea. I described that earlier. It's a drug produced by Regeneron. And the good thing about this drug is that it works. It's very clear it inhibits VEGF, and as a result, halts the progression of wet AMD. The challenge, however, is that it requires anywhere from 8 to 12 injections per year, because proteins don't last very long in the eye. And typically, patient compliance starts out high, but patients get a little tired of going into the clinical ophthalmologist 10 times a year to get a shot in the eye. So compliance begins to go down, and as a result, unfortunately, vision begins to degenerate and decay within these patients. So we had a very simple idea, which is that aflibercept works. So instead of delivering the protein, we genetically encoded it inside of an AAV and delivered the DNA encoding aflibercept. And in addition to aflibercept, there's actually a shRNA directed against another pro-angiogenic factor in the eye. Aflibercept takes care of VEGF-A and B, and this shRNA induces RNA interference against VEGF-C. So these are the preclinical studies, the IND-enabling studies, in which cynomolgus macaques were introduced into a study involving a laser-induced choroidal neovascularization model. So in this model, a laser is used to introduce mild burns at the back of the eye within the retina, and these burns induce the ingrowth of blood vessels, neovascularization into the region. And the delivery of anti-VEGF is known to inhibit the formation of these lesions. So this is a 6-month in-life study. It was a GLP study that served both for safety as well as for efficacy. So upon sampling vitreous fluid in the eye and subjecting it to an ELISA to quantify the aflibercept, you can see that at 3 doses, 10 to the 12th, 3E11, and 1E11, where 1E11 is a pretty small dose, detectable levels of aflibercept were found. And even at the smallest level, this is a concentration known to be therapeutic. We quantified the frequency of grade 4 CNV lesions. As you can see in the vehicle control, there's a high incidence of lesions. However, even the introduction of the smallest dose of the AAV was sufficient to completely prevent or inhibit the formation of these lesions. So based on these results, this product was taken into a phase 1 study early last year. And results, the second set of results were just reported out a couple of months ago. So within the first cohort, there were 5 patients enrolled, and these were high anti-VEGF need patients. So the way that this works is that patients go into the clinic, the clinical ophthalmologist tests their visual acuity by having them read letters on a chart. And if they've shown a decrease in visual acuity from the last visit, they get a protein injection because it's clear that they need a boost to be able to combat the wet AMD. These were high need patients who had a mean annualized injection rate of 11 injections per year. So you can see here for 4 of the patients, there was a lot of injections that took place in the lead up to the administration of the AAV. The 5th patient had a cataract and was not able to see, so they weren't able to fulfill this criterion of using visual acuity as a guide for the frequency of protein injection. Upon administration of the anti-VEGF AAV, these 4 patients have been completely off protein injection therapies ever since that time. And I'll remind you, the first patient was injected early last year. So vision has been stable since that time. Retinal swelling and edema, which is a hallmark of wet AMD, has actually been reduced. And so there's high safety and tolerability with no clinically significant intraocular inflammation or hypotony within the study. So I'd like to talk about a second study in which results were very recently reported out earlier this month. And this is a vector that was evolved for delivery to lung epithelium. So specifically, we took our library, aerosolized it, administered it to the lungs of non-human primates and homologous macaques, recovered the epithelial tissue, amplified the AAV that made it there, and repeated. And in addition, we introduced one further barrier for the virus to have to overcome, and that is neutralizing antibodies. Because as we discussed earlier, and as we saw in the prior elegant talk, pre-existing antibodies are a significant challenge in the population. So in particular, we took intravenous immunoglobulin, which is the pooled IgG from around 1,000 donors, and pooled and lyophilized into a single vial. And because most of those patients have been exposed to most of those donors have been exposed to AAV, that was a highly polyclonal source of antibody directed against AAV. So in this study, or in this evolution, we mixed together the library with the intravenous immunoglobulin prior to aerosolizing it and administering it to the non-human primate lung to force the AAV not only to have the ability to reach the lung epithelium, but also to evade the antibodies along the way. So the first target in where this vector was developed for a clinical trial was cystic fibrosis. This has, of course, been a long-standing target for gene therapy, in part because it's the most prevalent monogenic disease in the United States and Europe. There have been small molecule therapies that have been developed for this successfully, by Vertex, for example. However, there's still a fraction of the population that is not treatable with the Vertex drugs. So we took a mini CFTR, introduced it into the AAV, packaged it, and delivered it to lung. So I'll first of all show you the capacity of this vector to be able to evade pre-existing antibodies. Shown here are increasing concentrations of intravenous immunoglobulin added to the AAV. This is in vitro. And on the y-axis is the percent transduction. So you can see the natural serotypes are very rapidly neutralized. AAV8 is known to be, among the natural serotypes, the most resistant to antibody neutralization, as uniQure has found. However, this variant A101 resists higher concentrations of neutralizing antibodies compared to any of the natural versions. Upon aerosolization and delivery to the lung of non-human primates, you can see in this vehicle control, there's no immunoreactivity against the CFTR protein. However, in the case of the 4D710, the product encoding the CFTR, you can see high levels of expression in the trachea, bronchi, as well as alveoli. So again, boosted by this IND-enabling data, this was taken into a human clinical study, and these results were just reported out about 3 weeks ago or so. So in control human lung samples, you can see there is no immunoreactivity. This is CF immunoreactivity. There's no reactivity against CFTR. However, in biopsy tissue from 3 patients, you can see high levels of expression of the CFTR protein. And quantification of this indicated that 98% of the cells within these biopsy tissues were expressing CFTR. So expression was safe in that there have been no adverse events within patients. It's been very well tolerated. And furthermore, this latest data reported the first indications of function within the patients. So typically forced expulsatory volume, the amount of air that patients can expel under controlled conditions, decreases 2.3% annually. By comparison, within this small initial cohort of 3 patients, so this is obviously non-statistical, but 2 patients had a stable FEV and 1 had a 10% improvement in FEV over the course of the study to date. So to summarize, we have 5 products within human clinical development: X-linked retinitis pigmentosa, choroideremia, wet age-related macular degeneration, cystic fibrosis, and finally, there's a clinical trial ongoing for Fabry disease, a lysosomal storage disorder in which a lot of pathology takes place, unfortunately, in the heart. And this is using a vector that was evolved for delivery to the liver and heart. These data have been publicly reported, but 3 patients have shown significant increases in the expression of that enzyme, leading to therapeutic outcomes. So I'd like to summarize by saying that evolution works. Genetic diversification and selection can really be harnessed to solve complex problems in the gene delivery field. And we've utilized this approach over the past 25 years now to reprogram AAVs, to engineer AAVs with enhanced efficiency, targeted delivery, and the capacity to evade pre-existing immune responses. Vectors that we have engineered are currently being used in 8 human clinical trials, and the 7 that have reported data to date have all been positive. So we're currently implementing directed evolution to be able to address other challenges in gene therapy, including manufacturing, as well as regulation of gene expression. I'd like to close by acknowledging the people who did the work. The work in the mouse retina was done by Leah Byrne and Denise Delcara, each of whom has their own lab now. And that early work was done in collaboration with John Flannery at Berkeley. In addition, the work within the clinical work within 4D was the product of many people. I don't have time to acknowledge them all, but I'd like to acknowledge the CEO and my co-founder, as well as Melissa Cotterman, who came from my lab and was a co-founder of the company, and Peter Francis, who was CSO and CMO of the company. So thank you very much, and I'd be happy to address any questions when that time comes.
Thank you so much, Dr. Schaffer. A reminder to everyone that if you have questions, please drop them in the Q&A chat on the right of your screen, and we'll get to them at the end. And I will now introduce our last speaker, Dr. Weidong Xiao. Weidong Xiao is a professor of pediatrics at Indiana University. He serves as Associate Director of the Gene and Cell Therapy Program at the Herman B. Wells Center for Pediatric Research. He obtained his PhD from the University of North Carolina at Chapel Hill, where he started his research career on AAV vectors. He later held faculty positions at the University of Pennsylvania and Temple University before joining Indiana University. Xiao's research interests include molecular biology, investigating vectors for gene therapy, and developing treatments for hemophilia. So we can see your slides now, so please take it away.
Oh, thank you. I'm glad to have this opportunity to talk about my research on AAV genomics and its implications in gene therapy. Also, I was assigned to talk about preventing AAVs from integrating into the AAV gene into the host chromosome genome. But I think that is really a huge task. But here I'm going to talk about the mechanisms and see, potentially, maybe it has an implication in achieve this goal. Okay, so let's talk about AV species, AV genome species in the in a vector prep. And as you see here, yeah, as you see here, when we do a vector preparation and we run it on a gradient, we see, the empty particles and the full particles. And even when we got the full particles and we run it on gel, look at the genomes, and we see the huge, distribution of genomes from big to small. And so that shows, we're not get really a homogeneous population. So then, our lab, we developed a way, to map all the AAV genomes in a population, and we do this for many different preps. And we summarize all the genomes we found in our vector preparations. So we call them, one is, this actually was found like many years ago in the 1970s, and with people also showing, this kind of AAV genomes. They call it snapback genomes. So what the feature of these genomes is like, it's it has either the left half of the AAV genomes or the right half of the AAV genomes, but it's in the duplex form. So the AAV is kind of like this top strand and the bottom strand are linked together. And besides the typical, symmetric snap genomes, we also seen those snapback genomes that are kind of asymmetric. So it's not like in a way, we have a region that's like a duplex, and we still see a big loop. And we also seen this genome with deletions in the middle, and we see this in incomplete genomes or even more complicated genomes. So one of these things we try to understand is how this kind of like different AAV genomes came from. And to start this, and we especially look into the damaged AAV genomes. So in this case, like for when we make AAV vectors, and we typically transfect 3 plasmids together and into the 293 cells, and then we look at the AAV genomes packaged, and that's, and we usually expect, we get the genome we expect, just ITR and with the genome of interest. And in a special situation, we think about, what about, like in a situation that the vector genome was damaged, and it's just like, for example, just a small nicking. And then, we try to see this and say, well, like if there's a nicking here, supposedly this could be fixed rather easily, right? And then, we should still see the, full genome produced. But to our surprise, when we transfect the, this nicking the AAV genomes, and we see a very unusual situation. So to create the nicking on the AAV genomes, we take advantage of the CRISPR, nicking nickers. And because the nickers can either nick the top strand or the bottom strand, and so in this case, in the transfection, we basically, transfect the normal AAV vectors, and then in the presence of nickers or, in the guide RNA. So here is the experiment. Like when we transfect this plasmid with the CRISPR that will cut the DNA. So instead of like getting the 4 sides of AAV genomes, and we actually see 2 bands, like for example, it will cut in the middle, right? And then, we are actually seeing, either the duplex from the left hand, or, the duplex from the left hand, or the duplex from the right hand. However, when we only nick when we introduce the nickers, we'll either nick the top strand or bottom strand. We are seeing the corresponding band here. So if we nicking at the top strand, we are seeing this from 3' to this nicking side, we're seeing the 1.5 kb fragment. And when we see this, when we have the nicking, from the bottom strand, and we are seeing this, the 1.9 kb, predominant 1.9 kb fragment. So what it says here is like, when we have a damaged DNA, and we can really, seeing this duplex formation, so the snapback genomes are kind of like produced by the nicking or damage in the vector genomes. So there are 2 mechanisms that can be considered, lead to the formation of this like snapback genomes. One is this old, traditional theory that was long, proposed when the snapback genome was discovered for the wild-type AAV. It's called template switch. So what it means is that because of the DNA replicates, the polymerase error, And while they replicate AAV genomes, the polymerase will go from the top strand to the bottom strand, and this creates this point mutation. And because when we use this nickers, and we can show that this kind of some genomic particles can be also be formed, so we think that the DNA damage or NHEJ events could also lead to the formation of snapback genomes. So these are two possible mechanisms that lead to the formation of the subgenomic particles. And in another situation, we also looking into like in a situation that when vectors only have one ITR, and normally we think that when you have a vector plasmid, when you only have one ITR, we're not supposed to get any AAV produced. But actually, in this experiment, we did like a very unusual situation here. So one is like the control plasmid with no ITR, and this is the one that and we know that it will not have AAV vector produced. And then we have this vector plasmid with just like a single ITR, and we found like any vector with one single ITR. In this case, when we co-transfect the helper plasmid and adeno-associated plasmid, we can see that they replicate very well, even just with only one ITR. And for the vector produced, and we can even see this vector produced with a single ITR present. And so this clearly sees that when you have one single ITR, that's enough for vector to replicate and to pack AAV genomes. Then we look at the genome package from the vector with only one single ITR, and then what we found is that when you have a single ITR, even though the vector can still go on replication and packaging, but the vector population, when we look at the DNA extracted, and we see that the genomes are not uniform, there are usually multiple bands here. So it clearly shows that if you have only a single ITR, the packed genomes are kind of in heterogeneity, and so there are all sorts of vector produced. And then we run this. We use the DNA bioanalyzer to look at the genome distribution, and it shows the same situation. So it's just conform it. It just shows that if you have only a single ITR, and then the vector genome is kind of like is highly heterogeneous. Then people will ask why a single ITR species is a single ITR DNA is kind of relevant to what we are talking about, because most of people will report that when we have our vector plasmid for production, and we always have two ITR, and in most cases even when we have one like a missing ITR or some sort of mutations during the bacterial growth, the mucin ITR still retain portion of the ITR, and then because the AAV ITR can undergo a kind of like DNA repair, therefore somewhat in most situations this single ITR species doesn't exist. But people didn't probably what we need to do is we need to think about this. In this situation, most of people, most in our field think that the two ITR are always functioning in synchronized, but that's not the case, because in some situation, it's because of rep protein, in the vector production, and we have very low rep expression. So if only one ITR is utilized, so this creates a situation just like what we have modeled in the pre-study. So that it's kind of like a situation when you have one ITR, and that's going to create the heterogeneity, in the different DNA species we observed in the vector preparations. So I think I will summarize, what we learn about from the AAV genomics is that, one is DNA damage and the vector genomes are the primary reason for generating snapback genomes that create heterogeneity in the vectors we observed. And two ITR in the vectors may not always rescue in synchronization of for the optimized repeating expression that help AAV production. And the critical resolution size may lead to the production of snapback genomes in the AAV preparations. And the produced cells should be maintained well to prevent cell stress during vector production, because I think that's like when the cell is under stress condition, you got a DNA damage response, and that's going to create, like a problem for vector production. So here, I'm going to switch topic and talk about the biological property of snapback genomes, because the snapback genomes are showing here, like they can be small and long and short. So if you have like only the ITR snapback genomes, that could be one thing, right? This is pretty much like empty particles, because it's kind of small in a way, and it can be purified probably during the vector production, because the DNA content is so is not big. And then in the situation when we got the enhancers or promote only, and that could be a different aspects that we can talk about later. And another situation is that when you have vectors that will have the promoter and the partial genomes, and that's going to be created a different situation. And I will also discuss this one by one. So one of these old studies that like actually many years ago by Dr. McCarthy's group, and he showed that for self-complementary vector, and if he used only a self-complementary vector with just the promoter, as he's showing here, and when he injects this kind of like a promote only vector into the mice, in the specific strain mice, and it creates a lot of like tumors in the mice. So one of the problems that he looked at is because the vector integration, and this is a promote only, and that can activate oncogenes. But then let's look at the studies for this snapback AAV, self-complementary vector with promote only, and with this snapback genomes. So we can compare the difference. The snapback, the self-complementary with the promoter is like this group here. It's just a promoter and with a mutant ITR. And the snapback genomes from a regular AAV vector and with the promoter and gene of interest, that shows very similar property, like molecular configuration with the self-complementary vector with the promote only. So that's why this kind of like a vector could be problematic if you have like too much of this in your vector preparations. And then I think here we compared these two molecular configuration as shown here, the self-complementary vector with the promote only, and the snapback genomes that are produced from our daily vector preparations. And we checked, vectors are produced from like a normal triple plasmid transfection, and also vectors from purchased from commercial vendors that are made from baculovirus system. And we see, this kind of snapback genomes are existing in every vector preparations, and the amount varies. So this clearly shows this, in our vector preparations, and there is a potentially, damaging or bad particles existing in our vector preparations. So, and now in another studies, we also shows that, I think actually it's by Chen Wenli's group, and also by, that shows that, in the vector preparation, in the AAV vectors, and it shows the double-strand DNA formation. And the mechanism, the sort of was one of these mechanisms could be the, ITR function as a promoter that leads to the transcription of the negative strand. But I think that here, the snapback genomes can be utilized to as another explanation. So from a normal AAV vector expression, and we got the vectors, and this have the 6-strand DNA synthesis, and then we get the gene of interest expressed, and we get the protein expressed. However, when we look at the subgenomic particles, in this case, this is the subgenomic particles that has the promoter and the partial of the genomes. And this kind of subgenomic particles will have the, as you see here, it has an ITR. Then this ITR will undergo the 2nd-strand DNA synthesis. As you can see now, it has become a dimer. And in this dimer, then the positive strand and the negative strand are kind of connected together. And in front of this, it has a very strong, original promoter for transcription. So in this case, from this promoter and a strong promoter we use for direct gene expression, and we're going to generate a double-strand DNA. and this double-strand DNA can potentially, lead to the activation of immune response, and also, because this double-strand DNA is homologous to the transgene, so they could potentially promote the RNA interference. So the snapback genomes could, be like a negative influence the transgene expression. Then, we look at the aberrant transcripts. So the aberrant, the aberrant transcripts we defined here are, the transcripts with ITR and the negative strand RNA and the promoter RNA. And if we look at the AAV vector here, for example, we have the ITR and we have the continuous promoter, we have the transgene. But as you can see here, we're not supposed to have this kind of transcripts, the transcripts with ITR and the transcripts of the promoter by itself and it's a negative strand, because these are the normal vector we're using, right? And we're not supposed to see this kind of transcripts. However, in the older studies published by in Journal of Virology, and it shows that negative strand RNA can indeed exist in the wild-type AAV. And here, we inject the AAV vectors into the mice, and then at week 9, and we harvest the mouse tissue and in the liver, and we look at the, these are actually 3 different mice, and we look at the negative strand transcripts. And as you see, this is a vector that has the promoter and the transgene and also the poly-A. But this is all this data you probably are not going to see clearly, but as you see, we sequence the RNA, and we showed that the negative strand transcripts existing in all mice. So how can this negative strand DNA came from? And we then, we found that this kind of like a subgenomic species, the double-strand RNA can potentially play a role. And this shows the mechanism is called for AAV heterodimerization. It's utilized for AAV transplantation. In these 3 Nature Biotechnology paper, and the dimerization of AAV have been utilized to expand AAV capacity. For example, you have 2 vectors, and you inject them together. The 2 AAV vectors will dimerize together, and then they can help to expand AAV capacity. However, in our vectors, if we have this sub-DNA species, so this can be equivalent of a different vector. For example, in this case, it's let's say this is a subgenomic particles with a promote only, and this is another vector with a normal AAV genomes. Then, as you can see, if these 2 vectors can catamerize together, then we are create a new molecule that is not existing in our vector preps. So we have these vectors with the promoter, and it's followed by this promoter is the ITR, and therefore that's how the the kind of like a ITR transcripts can come from. And also, this has a promoter, right? And then this is under the control of the different the same promoter. So the transcripts, for the promoter can be produced as well. In a different situation, let's say this catamerization is, in the opposite orientation. Therefore, after this promoter, and you have the negative, the bottom strand, and therefore you can get the negative strand RNA transcript. So as you can see here, if you have this DNA subspecies, that's going to create a lot of problem for the unpredictable outcome for our vector delivery. So I would like to summarize, the potential subgenomic function in our vector preparations. So because, this subgenomic particle has 2 ITR, so it will behave the same way as normal AAV vectors in the host and target cell. So this will lead to probably second-strand DNA. It will undergo the second-strand DNA conversion, and it will do the concatenation, and it will integration, and it will, have all sorts of the same pathway as our normal vector do. So this will actually affect our vector transduction in many different ways. So it will probably help generate double-strand RNA, it will help form the neoantigen, and it will lead to the plasmid backbone transcription, and it will lead to the negative transcripts, and it will also undergo integration. Because I think for a lot of integration studies, and we have found that all the integrations are kind of like and in most situations we don't recover the full integrants. So a lot of times it most look like we have subgenomic particles integration. And probably potentially, we will have to promote the read-through, and it probably this will lead to cancer as well. So that's all negative aspects of these subgenomic particles. And then I'm going to talk about like how we can utilize the AAV genomics to design better AAV vectors. So let's use Zolgensma as an example. And for Zolgensma, and we know that it utilizes a self-complementary vector and to express the SMN gene. And as we know, like from the AAV population, and we have seen these subgenomic particles with this like with just one ITR. But as if you look at this conformation, these molecules are very similar to self-complementary vector, right? Because this has like the ITR in one end, and then the bottom strand and top strand are linked together. But the problem for this kind of molecule in the vector population is that it's not controlled, and so the generation of these molecules are random. So we cannot use it for vectors. So we found a new way of generating self-complementary vector by taking advantage of the snapback molecules. Instead of like let the vector generate this kind of snapback genomes randomly, and we intentionally use tail end enzyme to cut the vector genomes. So the vector genomes now, this enzyme has a special property that when it will digest the DNA, then it will link the bottom strand and bottom strand together. So in this case, we can create a kind of self-complementary vector that has a specific sites that the bottom strand and top strand are linked together. Then we can compare this with self-complementary vector. The self-complementary vector uses mutant ITR to allow the top strand and the bottom strand linked together. And this way, we have a tail end enzyme digestion that do this top strand and the bottom strand linked together. And this enzyme is highly efficient. When we digest this, we can make sure 100% of DNA can undergo this kind of conformation. But if we look at self-complementary vector, and by published before, and we know that it's about 50% or around 50% can have this kind of conformation when we do the vector production. Another way to think about the advantage of using this new way of generating self-complementary vector is like in this new CCE vector, we have 0 or no CPG sites in the ITR. And if we use a mutant ITR for making self-complementary vectors, there are still 16 CPGs in this region. So that's another difference of these two vectors. Then we look at the the vector purity and using this new way to make AAV vectors, and we compare the single-strand DNA vector, self-complementary vector, and the vector using CCE AAV vector without using the mutant ITR. And with self-complementary vector, we see that it's highly heterogeneous, and single-strand vector is under this situation is a single band. And with the CCE vector, we also see a single band here. And on the deletion gel, and it's even more clear. And with the CCE vector, without the mutant ITR, and use the new tail end digestion methods, and we get a very homogeneous vector population. And the self-complementary vector with the mutant ITR is kind of highly heterogeneous. And then we look at using this to compare it with Zolgensma, because in Zolgensma is made from self-complementary vector, right? With this promoter and the human transgene, and then we replace this mutant ITR with the CCE ITR, and then we look at the performance. And as you see here, with half dose of this CCE vector, and we can get better performance than the the full dose of the self-zolgensma. So this clearly shows that this our vector, with the CCE vector, has high gene expression under the same condition with the same promoter, same sequence. And this has been confirmed in multiple studies, and we use like a factor 9, and we use other reported gene, and it all shows that on the same situation, the CCE vector always have higher gene expression than the self-complementary vectors. So, well, finally, I'm going to summarize, what we learned about AAV genomics, and we think that the subgenomic particles in our vector preparation can be, lead multiple negative effect on transgene expression, because it may form double-strand RNA, and it may lead to the neoantigen formation, and it will probably lead to all sorts of like negative transcripts, and it could lead to the promoter read-through and potentially, cause cancer or whatever. And then, we can, utilize the what we learned about subgenomic particles, the especially snapback genomes, to design a better self-complementary vector for gene therapy. And finally, because still, we're at a very early stage of on the AAV genomics, and we can, if we know, we probably need to have more study on this, so we can have better understanding of the vector, the compositions, and that can be used to help gene therapy. And finally, I'm going to acknowledge everyone who have been involved in this study. These are included our lab members, and also the company started to, especially to develop the next generation of gene therapy products. And also, I want to acknowledge support from NIH and also the university. Thank you.
Thank you so much, Dr. Shao, and thank you to all 4 of you for participating today in the seminar. In the time that we have left, we'll get to questions from our listeners. So this first question is from Michael Schulman, and it's for Dr. Bowles. In brain-dead donors...
Louisa, are you there?
Yeah.
There we go.
Can you hear me? Can you hear me? Are you able to hear me right now or no? Can you hear me? Yes? No?
Yeah, I can hear you well.
Okay. So the question is for Dr. Bowles. So in brain-dead donors, will we eventually transfect donor organs with genes expressing tolerance molecules via catheters inserted selectively into their arterial circulation prior to organ removal rather than via ex vivo perfusion?
Okay, so it's an interesting question. I did not want to give the impression that the OCS was not being used for brain-dead donors. It is being used for both brain-dead donors as well as DCD donors. So I don't think that this would be something we would want to do, to administer the vector systemically to an organ donor at the time of procurement. One of the reasons is because one organ donor provides multiple organs: lungs, livers, hearts, kidneys. So getting accidentally getting getting a vector into these other organs might not be advisable to do. With that said, there are devices that are developed that are utilized are utilized for ex vivo perfusion of lung, liver, heart, and kidneys. So you could envision that perhaps these individual organs could be administered, their respective viral vectors using these organ-specific devices. And I guess another reason why I think we would want to avoid providing a vector to the organ donor in a systemic fashion at the time of organ procurement would be a safety concern. Perhaps the procurement individuals could be exposed, inadvertently to a viral vector. So I just don't think that that would be something we would want to do in this kind of situation.
Thank you. So this next question is for Dr. Kiani. It's from John Aletta. Do tolerogenic vaccines affect normal immune surveillance? And you're muted by now.
I just noticed. Very good question, and thank you for that question. I think the whole idea of developing tolerogenic vaccine is to tailor the immune response towards specific antigen while leaving the rest of the immune system intact. And the way that specifically we want to go about this by using both mRNA approach, which is transient, by using epigenetic editing, which is in essence tuning the level of the genes rather than bringing back to the homeostatic level rather than just hammering it down. We want to just limit the effect on the rest of the immune system, and by at the same time introducing the antigen of interest, we can just program the immune system to respond to that antigen. So it all goes back to spatial temporal regulation and the design of the formulation. Another design consideration, which is the physical chemical characteristic of lipid nanoparticle, is very important because they are designed in such a way that are not freely distributed in the body like small molecules. They are taken up by certain cells, including macrophages and dendritic cells. And so all of these effects happen in these cells. And so you kind of limit the effect systemically.
Okay. And we have a question for Dr. Schaffer. So how stable are the AIVs produced through directed evolution, and is there a risk that they will lose their beneficial properties?
Within the question, I'm not sure if stable refers to kind of the stability of the particle integrity themselves or the gene expression after delivery. I'll quickly touch on both. The particles are very stable and manufacturable. With every round of evolution, we're repackaging the libraries. And so packageability and stability of particles is a selective pressure that's built into the process. And as far as delivery goes, once the genome is in the nucleus of a cell or a cell within a tissue, it's at that stage, it's the double-stranded DNA is the final product, and the capsid really doesn't have any impact anymore. So it results in very stable expression, as we would expect from AAV.
Okay. Another question for Dr. Bowles. Wondering if you can comment on the use of AAV gene therapy for preserving organ function after transplant. Yeah.
No, that's a very good question as well. I think that it's not something we're actively working on, but I alluded to it in the context of the auto transplantation, where we could utilize those types of genes and improve function or repair the hearts and then reimplant them. So yes, I think that that's something coming down the line, but not right now. It's not the lowest hanging fruit.
One question for Dr. Kiani. So is there a way to predict whether a patient receiving AAV gene therapy will have an immune response to it?
That's a very good question. Generally speaking, I think this is an area of active research, and there are a lot of different factors that can go into forming the immune response against AAV. And I know that a lot of research groups or companies are looking into that actively. In general, with any sort of delivery of pathogens whether they're harmful or harmless pathogen, you expect the patients to form some sort of immune response to that pathogen, in this case, AAV. The extent of which and the duration of which obviously varies person to person, and the exact mechanisms that which drives that. So really, I don't know if it's completely understood. I don't know if David, do you want to add to that?
Oh, no, nothing more to add. That was
Yeah.
a great response.
Question for David. Can you evolve for multiple traits at the same time?
We effectively are. So if we take the lung example that I talked about, multiple traits are being pressured for. First of all, it has to be packageable, because of even if a vector is efficient, if it doesn't package well, it's going to be underrepresented within the library and kind of fall by the roadside. In addition, we mixed that sample with antibody in one of the selection steps. So it's being evolved or pressured for the capacity to evade pre-existing antibodies within those donor sera or donor IgG fraction from serum. And then it's being pressured for the ability to make it into the lung, because lung epithelial tissue was harvested and PCR was conducted to recover the AAV from that tissue. So that's probably our best example of multifunction evolution within the same study, in vivo delivery to an animal in the presence of antibody.
And a question for Dr. Kiani. So you mentioned that not knowing what the antigen is a problem for the reverse vaccination strategy. So what are some strategies to get around that or to solve that problem?
Well, in the case of, I mentioned that mostly in the case of reverse vaccination for autoimmune diseases, for transplantation, where, the nature of the immune response that happens for transplantation or autoimmunity, we call it very polyclonal. There are a lot of antigens that can cause T cell response to happen. In case of gene therapy, we are kind of more fortunate because viruses are, we know their, antigen repertoires. And we also know when we can easily identify the antigen repertoires of, the transgene that they carry. And when, there are a limited number of antigens to play with, it's kind of like easier to induce tolerance. And with transplantation and autoimmunity, again, there are a lot of work that some researchers are doing using machine learning and AI to be able to predict the antigenic epitopes of the organs or different conditions. But usually those give up a list of antigens, major antigens, minor antigens, that somehow it's difficult to produce in the form of peptides in larger scale to create these vaccines. However, I believe that advances of mRNA technology, whether it's limited number of antigens or is it more than antigens, will kind of help us overcome that kind of manufacturing challenge, because you can encode these with an mRNA or even DNA and deliver and use cellular factory to make these peptides. With gene therapy, we are more fortunate because, as I said, there are, limited number of antigens. In our case, for instance, if you just deliver the whole capsid of the virus in a form of mRNA and let the cells process that and present whatever antigen they want at their surface.
I just wanted to interject for just a second for folks. I'm sure lots of folks have been already noticing this, but FDA just approved BioMarin's gene therapy for Heme A within the past hour. So FYI, good news for the field.
Good. Wow.
What is that gene therapy for?
Hemophilia A.
Oh, hemophilia A. Okay. So this question is for all four of you, and maybe we can start with Dr. Shao. How do you see your research impacting the future of gene therapy?
Well, that's a big question to answer. I think for my research, I think it's from a different perspective, because if a lot of research has been focused on capsids, and we look what's inside the AAV and look at how these genomes will affect the long-term gene therapy, because like for initial transduction, and I think everything is probably more dictated by the capsids because it's directly where the vector genomes will go. But after everything's settled down for long-term gene expression, it's all on the AAV genomes, right? So if you therefore we have to control the genetic composition. We don't want something unexpected or too many of those in the vector population, so that will affect outcome. So I see that we will have to design new ways to improve the vector components, and we have to also to think about how we can utilize those components to design better vectors. I think that's how I think that the future of my research will on gene therapy.
Dr. Bowles, would you like to go next?
Sure. So, right now for advanced heart failure, cardiac transplantation remains the gold standard treatment. But it's really unfortunate that for most people, these hearts last on average about 11 years. And not only that, but one of the, primary concerns that they're living with is long-term immunosuppression, which can lead to cancer, can lead to kidney failure, can lead to organ, failure of the organ they've received. So I think, at a very minimum, my work could extend the life of these patients who have had heart transplantation and give them a better quality of life. So I think if that works out, I think that would be where the contribution would lie.
Dr. Schaffer, would you like to go next?
Sure, thank you. So we've really been pursuing the idea that gene therapy, of course, is, as we all know, very complicated, and it's in that environment where it's a complicated set of molecules that have to carry out very complex steps to be able to make it from a syringe into the nucleus of a cell. We have believed that high throughput approaches in screening and evolution is a very powerful approach where the field is going to continue to advance our basic knowledge of how this virus works. We don't necessarily have to wait for those advances in order to rationally design viruses. We can evolve and be able to create highly effective, targeted, efficient vehicles without having to wait for the outcomes of basic studies. And in some ways, investigating the results of an evolution can teach you something basic about the mechanisms of virus host and virus cell interactions after the fact. So we're hoping that we're making contributions to overcoming delivery, which, as we all know, is a significant barrier in the field. And we again think that high throughput screens and forward genetics are going to be effective approaches for tackling the next problems that are coming up, such as now that we have 5 FDA-approved AAV gene therapies, we're going to need to manufacture a ton of vector. So that's a next problem that I think should be of strong interest to the field.
Go ahead, Dr. Kiani.
Yeah, and for me, I think along the way, I have been very inspired to somehow address the issue of access to gene therapy and how can it be available for the patients who really need it at the right time. And I think throughout years, my research have been somehow trying to address that. With this particular work that I presented here and we are pursuing at GenXGen, the hope is to, by instructing the immune system to ignore gene therapy viruses, we'll have these life-saving therapies accessible to those patients who are already excluded from clinical trial because of high pre-existing antibody. And there are a lot of those patients, about 40 to 50%, I mentioned, are excluded because of that. So that's the hope. And we know that's a very big challenge to tackle. It's a very hard problem to solve. But that's a part of, approach or the path that I'm personally very inspired to pursue. And the other thing that I'm hoping that all of us here, and I'm sure all of us are thinking about it, is the price tag of these gene therapies, which is something that, by advancements that happen, like for instance, David's work in manufacturing and other companies that pop up in the field, hopefully with the right amount of work, we can decrease the cost in our ways, that in our particular way of thinking about decreasing the cost, is about going after mRNA approach, which is cheaper than AAV manufacturing. At the same time, picking genes such as MYD88, which are implicated in a variety of different conditions. So hopefully, once you develop it for one purpose, you can repurpose it for other situations, and therefore kind of like think about ways to decrease the cost. So that's my thought process here.
Yeah. So one last question for all four of you. What are the main challenges left in AAV gene therapy research? And Dr. Schaffer, you had mentioned manufacturing. Would you like to add anything else to that?
Yeah, I think delivery is still, there are a lot of unsolved problems in delivery, right? There's not a great vector that reaches skeletal muscle after intravenous administration. Just, one example. Manufacturing is a significant challenge, especially as the field, hopefully increasingly heads from trying to help out patients who are suffering from rare diseases to patients that are suffering from diseases that affect large numbers of patients. So, have to manufacture a lot more vector. And then in addition, in the early phases of gene therapy, we've been treating conditions that have a broad therapeutic window, right? Where delivery of, less than natural levels or overexpression way above natural levels of a protein doesn't harm a patient, because, you want to start simple, as simple as possible. So, hemophilia B, you can overexpress, several 100% of normal levels of factor IX, and that's okay. Whereas a field can head towards conditions where it matters when, where, and how much of a therapeutic RNA or protein is expressed. So we're going to have to, in the future, start worrying about really complex mechanisms to control gene expression and regulate it well. So those are, the future challenges that I kind of foresee for the field as a whole.
Dr. Shao, would you like to go next?
Oh, well, I think for gene therapy, the promise is you are supposed to quickly develop another pep plan based on what we learn about the vector. And supposedly, say, well, you have this capsid, you have the vector, you are supposed to only need to replace the gene, and this vector should be good enough, and to treat another patient. And that's how the promise of gene therapy, because it's so supposed to be very predictable. But until now, we have seen look at every indication is challenging, and we see we have to spend a lot of time to develop this. So this creates a huge problem for making gene therapy to fulfill the promise of gene therapy. So I think I think the challenge is we still need to understand the AAV vectors and a lot more than what we know today. So the vector can be really simple in a way that we can replace every transgene, and we can treat the patient with just single, because a lot of the rare disease, we don't have a lot of patients, right? So in this case, the gene therapy can be used in those patients relatively easily.
Dr. Walls?
I really don't have that much more to add between what Weidong and David have said. I might want to just end to sort of tout the delivery system that we've developed with the ex vivo perfusion. I mean, we've overcome, I think, a lot of the challenges with the in vivo delivery. Moreover, I think we can deliver cocktails, of many different gene viral vectors with many different genes. So I mean, I think that our system, is exploitable. It's just again, it's a smaller patient population, but it's still, I think, a prevalent patient population. And again, I think the Achilles heel is manufacturing. I think that's really what's going to be limiting us at the end of the day, is how much we can manufacture for all of these applications that we're developing.
And I want to add, in addition to all of the things that was mentioned, which I agree with all of them, I want to kind of take another, bring in another perspective, and more from the patient's perspective. And that is, these gene therapies are life-saving for a lot of people out there. Unfortunately, though, the advancement of gene therapies are very slow as opposed to, what patients need. So I know from technology development, there's a lot goes into developing this technology and the process that we need to take for FDA approval. But this is more like a question that I don't know the answer of, but something that I constantly think of, how we can accelerate the process of development while reduce cost. And part of it kind of comes back to the fact that how can we increase more competition in the field and address some of the limitations that would not help us in the process, including like some of the regulations that we need to overcome that can be very lengthy, some of the IP issues that sometimes are not helping, rather they are, providing roadblocks for innovation. And this is a bigger type of questions that are very important to at least be on our minds when we are developing these therapies for patients.
Well, thank you for that. And unfortunately, that's all the time that we have for today. So I want to thank our 4 speakers for their wonderful talks and for sticking around to answer questions. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the seminar will be archived on the DDN website, and you will receive an email notifying you when the webinar is available on demand. On behalf of DDN, I would like to thank our speakers, Dr. Bowles, Dr. Kiani, Dr. Schaffer, and Dr. Xiao, as well as our sponsors, BPS Bioscience, DNA Script, and Biorad. And of course, thanks to you for listening. Goodbye. Have a great afternoon.